Hello, I'm Jacob Plant, and I'm here to talk to you about phages. I know this thing looks a little weird, but we're going to get through this together. Now, I think it's safe to assume that most of you have at least heard of antibiotics, even if you've never taken them. What you may be less aware of is the risk posed by pathogens that become resistant to these medicines. These antibiotic-resistant bacteria are a very real and dangerous public health risk, as they can be harder to treat and have much higher mortalities than their non-resistant counterparts. In 2019, the CDC estimates that there were 2.8 million cases of resistant infections in the US alone, resulting in at least 35,000 deaths. Since it could take more than a decade and billions of dollars to develop new antibiotics, while resistance can arise spontaneously, it would seem prudent to research alternative ways to combat these pathogens. Enter phage therapy, which uses bacteriophages, or viruses that target only bacteria, to treat bacterial infections. In order to explain the merits of this treatment, I want to share with you the effects of phage therapy on their own, as well as how they compare to more traditional antibiotic treatment. Then, I will talk about the downside risks of using phages medically and other potential curatives for the antibiotic-resistant crisis. By the end, I hope to have convinced you of the benefits of development of phages as a medical tool. Though people had been using antibiotic substances before this, the first true antibiotic, penicillin, was identified in 1928, and they had entered widespread use in medicine by the 1940s. New antibiotics were and are still discovered over time, but not faster than the targeted pathogens can become resistant. Some antibiotics can remain viable for decades, while others become resistant in less than a year. The use of phages would require a separate form of resistance that most bacteria don't easily accommodate alongside antibiotic resistance. This would allow for new, effective treatments that can even be safer and more efficient than antibiotics, even for non-resistant strains. Phages themselves are simple. Compared to the cells they infect, though, they are varied. The two components you need to know are the genetic material they inject into their host and the enzymes they use to attack and kill their host. When the phage enters their cell, there are a few pathways it can take, but the one we're interested in is the lytic pathway. This is probably the most direct path to killing the cell, and it involves the phage tricking the bacteria into making copies of both genes and other components, as well as other substances that will break open the cell. This is called lysis. As the process is being carried out, two classes of enzymes are produced, holins and lysins. These act as a sort of clock on the entire pathway, as holins will create pores in the bacterial cell's membrane, and the lysins will move through these pores to break apart the cell wall. These two structures, the membrane and the cell wall, are key components of the cell, and breaking them open results in the internals of the cell and the new phages leaking out, killing it, similar to poking a hole in a water balloon. You may be wondering, what happens if these phages infect me? The long and short answers are, they won't. Phages are very specific in their hosts, and many will only infect a single species, or even a single strain of bacteria. They are also only infect bacteria outright. Though they are similar to viruses that infect humans, they will harmlessly interact with our cells. In case you wanted proof, phages are a part of a healthy human microbiome, just like the bacteria they might infect. This is the basis of phage therapy, administering a phage cocktail to a person or at the site of the infection and allowing the phages to clear out the offending pathogen. Fortunately, other people have already done the hard work of compiling case studies and trials concerned with phage therapy. Liu et al. Reviewed a total of 69 reports on phages using an anti-infection role, and they found that the effectiveness of the treatment depended on how sick the individuals were at first and alongside how the phages were administered. This was based on a combined 49 clinical trials and case studies with humans, and general observations were drawn from another 20 animal studies. They found that in individuals who were first not sick and second where phages were administered orally, the phages passively passed through their GI tract and passed in their feces. While when injected into the tissues of the patient, an immune response was generated quickly, and this resulted in the production of antibodies being formed, which attached to the phages, preventing them from infecting anything. They did note that there was the possibility of endotoxins, which are a part of some bacteria that can trigger an immune response, being released from the bacteria when lysed. Overall, they determined that in the human subjects, the phages were well tolerated, and most patients improved by the end of treatment, if they didn't fully recover outright. In terms of how phage therapy compares to traditional antibiotics, Lin et al. Found that several notable differences. First, overall immune responses tended to be reduced against phages, although this might have been how antibiotics can be sequestered within tissues, while phages will normally just stay out. This simply reduces the risk that a patient's immune system will reject the treatment. And second, whereas antibiotics can pose a risk to the person as they will affect any cell that can take them up, phages are highly specific for a narrow range of hosts, and only for bacteria. This is also simply another safety measure, as the patient is less likely to be harmed by the administration of the phages. The degree of danger a potential response could pose is not particularly well known and is disputed due to the low number of well-controlled experiments. Third, phages can kill bacteria that have formed a biofilm. These form when some quantity of bacteria exists in a single spot and begin to cover themselves with a slimy matrix. This makes it very difficult for immune cells to affect them in any way. Antibiotics are not able to readily penetrate this matrix, but many phages have enzymes that allow them to move through and get at the bacteria within. Biofilms are particularly nasty when they form on implanted surfaces, such as artificial joints that you might find in elderly patients who already have weakened immune systems. The last comparison they made was of the costs associated with preparing the treatments. It turns out that securing and growing the phages, then purifying a large enough dose can be quite expensive. A standardized cocktail could be used to reduce the costs, but it may not be as effective as tailoring a dose for each individual patient. Aside from the potentially prohibitive costs, which may not be an issue with the development of better techniques, and which is not necessarily accepted as an issue by everyone, Locario and Abaddon found that not all phages are viable for use in treatment. For a phage to be considered, it must follow the lytic pathway only, it must be able to be stored under reasonable conditions, and it would be best to confirm that none of the phage genes code for toxins, among other restrictions. Contributing to this is also one of the potential benefits, the specificity of the phages. If the phage used isn't able to infect the desired pathogen, it simply does nothing. The administration of the wrong phage could also have a negative effect on the patient's microbiome and have downstream effects later on. Of course, the immune responses triggered by phages must also be monitored, as a severe response could make a patient's conditions worse and lead to their death in severe cases. The difference in effect between immunosuppressed/compromised patients and their immunocompetent counterparts is not well understood, but there is some textbook evidence that supports either type of patient having a better response for different reasons. And while some of these issues are not particularly insurmountable, others, like the potential cost if technique is not improved, are more troublesome. As are the circumstances, alternative methods to handle the antibiotic resistance crisis might seem more appealing. Ventola found that the biggest contributors to the speed at which new resistances arise is the inappropriate prescription of antibiotics for people who don't need them, and the extensive use of antibiotics in agriculture. Both of these can create circumstances that spur the development of resistant strains without a patient ever being sick at first. Ventola proposes that more strict requirements for the prescription of antibiotics for non-serious infections, as well as reduced usage overall, could be the remedy we needed. Additionally, reduced barriers to the development of new antibiotics, including things like government grants and a review of the regulations in place to moderate any development, could also create new opportunities for antibiotic development. A quick summary before we move on. So far, we've seen that use of phage therapy that has been documented shows positive results, and phages have some advantage over antibiotics, with many of the downsides being logistic in nature. There are also alternative ways to begin fixing the antibiotic crisis, most of which involve stopping doing what is contributing in the first place. I want to clarify that the development of phage therapy as medicine counterintuitively doesn't have to result in phage therapy itself. As we've seen, there's a real risk for an immune response which can both harm the patient and reduce the effectiveness of the treatment. As a potential solution, I want to recall your attention to lysins, one of the enzymes that actually break open the cell. Lysins, unlike the whole-in enzymes, can lyse the bacteria on their own and may be better than whole-cell phage therapy. This could be the case for three main reasons. One, lysins are smaller than phages, so they may be less likely to be picked up by immune cells and cause a response. Two, it's easier to engineer individual proteins compared to whole phages. And Lin et al. Reported that the addition of a domain or protein region that can insert itself into the cell wall increased the effectiveness of the enzyme in saving mice from MRSA, a methicillin-resistant bacteria. Third, the method by which lysins kill cells is particularly difficult for bacteria to become resistant to. One part of this is the critical nature of the cell wall to bacterial species, so changes are more likely to be damaging than beneficial. And second, part is that trying to become resistant to this would need a completely unrelated change in the cell wall of bacteria. because this form of treating an infection would not leave resistant cell behinds because it is 100% lethal, as the viral components would now be outside the cell. Notice that the end results of using lysins and phages are the same, but adding the step of infecting the cell for phages. This step is critical in the difference of long-term effectiveness between using lysins or whole phages, as it gives an opportunity for the target pathogen to develop some novel resistance. With how much I've been touting phages as the next big step in medicine, it might seem like reversing course to say that using a combination of phages or lysins and antibiotics is actually what works best to clear out infections. Allow me to explain. For every antibiotic resistance a pathogen has, it must expend some amount of space in its genome, as well as some resources to carry out this resistance, whatever its mechanism. The same is true for resistance to phages, but it is, quote, less efficient because phages' method of killing cells is more complicated, and the space required in the genome for this resistance is larger. The effective result is that a bacteria would need to forfeit parts of its antibiotic resistance to successfully resist phages. If that happens, then they become vulnerable to the same antibiotics being used, which can kill them outright or weaken them enough to let your immune system deal with them. So what's left before we start using phages or lysins in the medical theater? Well, we need more controlled trials with humans to better understand how our bodies might react. Coincidentally, phages identified in humans don't seem much like what we find outside our microbiome, both genetically and physically. The specific factors we need to better understand include how dosages and toxicities are determined, how does endotoxin release affect the surrounding tissues, and how to reliably purify the cocktails of contaminants. I think that all of this is easily doable once resources are directed for the development. Most of the roadblocks would need to be faced by basically all modern medicines as a standard part of the regulations required. While I myself think that the use of lysins will be more practical than whole phages, to do either, we will need to get a better understanding of how the phages themselves work in this medical context. I believe I've shown that the foundation for phage therapy is ready, and that it has real potential as a solution to the threat posed by antibiotic resistance.